Hello and welcome. My name is Victoria Rees and I'm the Deputy Editor of Drug Target Review. I'm joined by Arun Srivastava, George H. Kitzman Professor of Genetics and Chief of the Division of Cellular and Molecular Therapy in the Department of Pediatrics, Department of Molecular Genetics and Microbiology, and the Powell Gene Therapy Center at the University of Florida. Arun will be speaking at Drug Target Review's Cell and Gene Therapy Advancements Online Summit on 3rd to 4th of November this year. Click the link below to reserve your spot and hear about Arun's research on next-generation AAV vectors. Today, Arun will be discussing how to combat the immune response to AAV vectors. But first, let's get to know him. So Arun, would you be able to give a brief background to yourself?
Yes, hi, Victoria. First of all, thank you so much for giving me this opportunity to talk about some of the work that we have been doing. And by way of very brief background, this is my 41st year of having worked with AAV and AAV vectors. So I have been at it for a long time. And I think I've seen from the very beginning, the ups and downs, mostly downs of gene therapy. But I'm hoping that, some of the work that we have done in the lab, hopefully will help the patients tomorrow.
Definitely. Thank you so much. Well, of course, I want to ask you about your research, but first, I'd like to just ask you a bit more of a general question. Why do we need to develop gene therapies?
That's a very good question. So there are more than 6,000 human genetic diseases that we know of, and it is very difficult to find cures for each one of them. So one idea has been to try to replace or correct the defective gene in the body. And that's why I think gene therapy, which actually has been talked about for more than 2 decades, but looks like it's finally beginning to show some promise.
So how exactly do AAV vectors work and what role do they play in gene therapies?
So AAV is a very unique virus because it was discovered in 1965, and today it has not been associated with any known human disease. So this is the only virus that causes no disease. So a few years ago, it became very obvious that this could be used as a very useful vector to deliver genes back into cells and animals and now in patients. So AAV has turned out to be a very efficient vector for that purpose. And now that we have 2 FDA-approved drugs, so it has really seen a renaissance in a sense that initially AAV was not considered very useful because it doesn't do anything. So people used to make fun of this virus. They thought AAV stood for almost a virus, meaning that it doesn't really do anything. But since now we have, at least 8 human diseases have been cured, or at least have shown clinical promise with AAV vectors, the new name for AAV is an awesome vector.
Definitely a very apt name there. So I wanted to ask, why can they prompt a response from host immune systems?
Another very good question, Victoria. So remember, this is a virus, which means, most uninfected people have never seen this before. So it is, in essence, a foreign protein. In other words, it's actually an antigen. So anytime you inject a foreign protein into the body, it will mount an immune response. Body will mount an immune response to get rid of it, right? So what is amazing about this virus is that it actually it's fairly mildly immunogenic. So it actually does not cause significant problems. The problem has been that lately all these biotech companies have been now injecting massive doses of this vector into patients, and beyond a certain limit, the immune system simply gives out. So just to give you a very quick example, there are 30 trillion cells in human body, in an adult human body. So AAV actually can be given at a dose 3 times the number of the cells in the body. In other words, you can actually inject up to 100 trillion AAV particles, which is like one. For each cell, you're giving 3 AAV particles. And in some cases, it has gone up to 42 times or 700 times. So you can imagine, too much of a good thing can be, bad, right? So yeah, within a reasonable limit, I think, AAV vectors have been shown to be fairly safe.
So of course, your research has been into AAV vectors. Could you just give a bit of detail about your work in the next generation AAV vectors?
Yes. So about 20 years ago, I had this epiphany, if you can call it, is that, AAV actually, I think all of us can agree that evolved as a virus and not as a vector, right? So when AAV first infects us, within 1 week, our body will mount an immune response and clear the virus. So when we use the exact same virus protein to deliver our genes, the immune system has no way of distinguishing it from the naturally occurring virus. So therefore it attacks and tries to clear it. And that's one reason actually people are going to higher and higher doses to overcome the immune response in the first place. So we decided that no, the vector has to be different from the virus. So that's led to the development of what we are calling the next generation of AAV vectors. And without going through all the technical details, this was all published work. We published this in 2008. So basically, we made a few minor changes on the capsid surface. So now the host immune system does not see it. So now the virus can get in, or the vector can get in and deliver its payload or the gene of interest to the cell. And therefore, it is much more effective and much less immunogenic.
So you've had some really promising results from that then?
Yes, yes.
Fantastic. Well, aside from the successes, what challenges have you faced during your research? But then how have you overcome some of those challenges?
Another very good question, Victoria. So as I mentioned, 40 years ago, no one took this virus very seriously. So it was impossible to get research funding. So because they say, no disease meant only one thing, no money. So, so it was quite a bit of challenge to get, research funding to do work on AAV. But I decided that, as a biological system, this was interesting enough for me to, continue to work with this virus. And therefore we were able to develop, as we learned more about the basic science of the biology of the virus, we were able to develop the next generation of vectors. So I should mention, perhaps this will come up later. So one of the vectors we created in the lab actually the next generation vector has gone into a phase 1/2 clinical trial for a disease called Leber hereditary optic neuropathy. So I should also very clearly state that I'm not a clinician. So the trial was not done by us. It was done by our collaborators and colleagues at the University of Miami. And they injected the vector that we created in the lab into the eye of 14 patients, and 13 of these 14 patients actually regained their vision. So something that we did in the lab actually has helped patients in real life. One patient, it did not help because his disease had progressed beyond the treatment window. So I feel good about that whatever we have been able to do in the lab has actually helped real patients.
Definitely. And it's, your research actually in action, which must be so fantastic to see. So what about actually the next steps for your research with AAV vectors?
So we are working on, beyond the next generation, we are now working on what we are calling, for lack of a better term, third and fourth generation vectors. So the idea here is to do two things, hopefully accomplish two things. So as you know, no two humans are alike, right? So our hope is to be able to develop what we are calling the patient-specific AAV vectors. So we, see, a good example here is that, two patients come up with the exact same cancer, let's say, for example, and you give them the exact same drug, and one patient benefits from the treatment, the other doesn't. And so same thing is perhaps happening in gene therapy, that, same vector that is being given to two patients may not work as efficiently in both patients. So we are trying to figure out a way to how to make AAV vectors patient specific. And the other, which I'm sure everyone knows about, a very interesting area that has recently been developed is genome editing. So our hope is that we will be able to achieve in vivo genome editing. So deliver vectors and let the vector do the job without having to isolate cells, do manipulation ex vivo, and then replace those cells back in the patients. So if we can do that, I think this would turn out to be a very simple, single injection hopefully can cure human disease.
Fantastic. Well, hopefully we'll be seeing some very positive results from that soon. But I just want to say thank you so much for speaking with me, Professor Arun Srivastava, and for giving such excellent answers. It's been an absolute pleasure to speak with you today.
Likewise, Victoria, thank you so much for the opportunity, and I look forward to participating and discussing future possibilities at this symposium that you're organizing. So I'm greatly, greatly appreciative.
Yes, definitely. And also, thank you for watching. If you would like to hear more about Arun's research, do indeed join Drug Target Review's Cell and Gene Therapy Advancements Online Summit, where you can learn more about combating the immune response to AAV vectors and more about his research. Click the link below to make sure you do not miss out, and we hope to see you again soon. Goodbye.
Goodbye. Thank you.